Peace be upon you all, and welcome to a new exciting session of the Seeing God in Biology series with Dr. Rana. And this is essentially the second installment of the central dogma of molecular biology. Faz, how are you doing today?
Ahmed, I'm doing great. How about you?
All is just well. So first, let me put my headphones on. But you're going to talk to us today about the first step of information flow in the central dogma. Is that correct?
Yeah, that's exactly right. Because again, um, the central dogma is all about the flow of information, and one very important aspect of that flow of information is the replication of DNA. DNA is the really the data storage system inside the cell. It's harboring all the information that the cell needs to make proteins. And so when that cell divides, that information has to be transmitted to the daughter cells, and that's through the process of DNA replicating itself. And as we'll come to see, that there is a logic to the structure of DNA that lends itself to being replicated, but also that there is a logic to the process of DNA replication as well. So it's a complex process, but when we ask that famous why question,
Why?
yes,
Yeah, we will find an answer to that too, right?
yes, we will.
let's have a dive into it.
Sure thing. Well, here's a cartoon showing the structure of DNA, and we've shown this cartoon on several occasions. And what I want to do is just emphasize the parts of the DNA structure that really lend itself again to being replicated. And DNA has a ladder-like architecture, where the backbone of the DNA molecules are like the uprights of the ladder, and the side groups that interact are like the rungs of a ladder. And really, it's two molecular strands of DNA that come together that interact to form this ladder-like architecture that if you twist, you get the well-known DNA double helix. And there are four subunit molecules that are used to build DNA. These are the genetic letters. They are abbreviated A, G, C, and T, and it's that sequence of genetic letters that contains the information in DNA. And as it turns out, that the two strands of DNA to first approximation align in a parallel manner, but in actuality, it's an anti-parallel arrangement where the top of one strand aligns with the bottom of the other strand. And the top and bottom designation for biochemists is with the nomenclature 5', 3', 3', 5'. So there's an orientation to the DNA strands. There's a polarity to the DNA strands that is, again, exemplified with that 5', 3', 3', 5' nomenclature. And we're going to discover that this anti-parallel arrangement is something that the DNA replication process has to be able to manage. And as we talked about in a previous episode, that anti-parallel arrangement is actually critical because it gives stability to the double helix, which is important in terms of its role as an information storage system. And it also creates topological features in DNA in the double helix that allow for protein binding, which is also very important, not only for information storage, but to access the information and to replicate the information. Now,
just to be sure of something, essentially, if we have one strand, the other strand is like, in the old days of photography, you had the negative of the image, right? So regardless that they are running anti-parallel, but essentially, if this one is going 101, the other one is going 010.
yes,
It's not like one is written fuzz FUZ and the other one is written ZUF. No, it is just that the F has the negative image of it at the other end. So somebody can actually read the information from this one or that one if he takes notice that this one is just the negative of that one or the complementary of it. Is that correct?
That's exactly correct. And this again is a very important feature that allows for DNA replication to take place, because as it turns out, there's a set of rules that dictate how the side groups of the DNA strands interact with each other. And this is
Uh-huh.
called the Watson-Crick base pairing rules, where
Mm.
the G, or the guanine, on one strand always pairs with a C or a cytosine on the other, and an adenine or an A on one strand always pairs with thymine or T on the other strand. So if you know the sequence of genetic letters on one strand, you're going to know the sequence on the opposite strand because of these complementary base pairing rules.
Mm-hmm.
And
Mm-hmm.
so in this particular cartoon, it's emphasizing this point. And you can notice from the cartoon how the different strands are oriented, again, in the opposite direction, right? Uh,
Yeah.
if you look carefully at the backbone, but you see that C is always pairing with G, T is always pairing with A. And so the sequences of those two strands are complementary. Now, what's interesting is that when Watson and Crick in 1953 in Nature published their uh, their structural analysis of DNA, where they proposed the structure of DNA, they have a sentence in the paper that is a very understated, where they said the structure immediately suggests the means by which DNA is replicated. And
Wow.
so the way to think about this is that if you could conceptually pull the two strands apart, and then each strand now serves as a template that then allows you to assemble the complementary strand. And what
Uh-huh.
would result would be essentially two identical molecules to the parent molecule. And so biochemists call this uh, template directed semi-conservative DNA replication. And what they mean by template directed is that each of the strands serves as a template to assemble a complementary strand, and that the resulting daughter strands have an original strand from the parent and a newly synthesized strand. So it's
Ah.
semi-conservative, uh, template-directed DNA replication. Now, what's interesting again is that this mechanism of replication literally falls out of the structure of DNA. So it turns out, again, that the complementary base pairing rules is why DNA, the information in DNA, can be replicated. Uh,
Mm-hmm.
and and again, it really demands that the only way for replication to take place is through this, again, template directed semi-conservative replication. Now, what's interesting is that prior to the discovery of the structure of DNA, biochemists learned that DNA was the genetic material. It was the molecule that stored the genetic information, but they didn't know how it would replicate. So, so there were three models that people were uh, exploring. One was the semi-conservative model. The other was what's called a conservative model, where you have a newly synthesized molecule and the original parent molecule regenerated. And then the other model was a dispersive model, where pieces of the two strands and the newly synthesized strands would be all kind of jumbled together. But it turns out that it's a semi-conservative, replication model. But again, there's an elegant logic that undergirds, again, that means of replication. And so I think we don't want to under appreciate how
Mm.
how the structure of DNA really, again, is ideally designed not only to be stable so that the information can be stored with integrity, but it simultaneously is designed in such a way so that replication is possible. And
Wow.
this is a problem confronting the RNA world hypothesis, which is one of the leading explanations for the origin of life. And in a past episode, we talked a little bit about some of the intractable problems with the RNA world's theory. Well, one of them is, if RNA is both the information storage system as well as the catalytic molecules that are responsible for carrying out all the operations in the cell, you have to replicate RNA. And that's a big challenge, is nobody's been able to produce a truly self-replicating RNA molecule. And
Wow.
and yet with DNA, the subtle differences between DNA and RNA actually lend themselves to lend the DNA molecule to actually being a bona fide self-replicating system. And
Yeah.
so again, we don't want to underappreciate how remarkable that feature of DNA actually is.
If I understand correctly, what you have on the screen here, which are the three possible models, obviously the one to the very right, which is the dispersive model, would be a complete chaos because you wouldn't know which part is coming from where. The one in the middle, the conservative model, just my first impression is that after the first replication cycle, the blue one and the pink one, for example, if an error happens during replicating the two blue ones, then both are copies and you wouldn't know which strand is the correct one and which is not. But if it's the semi conservative one, each copy has an original and a copy. Each daughter has an original and a copy. So if there is a problem with the copy, you can, in the new cell, refer to the original, or actually before the new cell maybe is formed, you can refer to the original to make sure that the whole daughter DNA two-stranded molecule has been faithfully copied. Is that one of the benefits of the system?
Yes, it is. Yeah, that's a really insightful point. Is that the semi-conservative approach actually lends itself to high fidelity, ensuring high fidelity replication, which is absolutely critical.
Yeah.
It's absolutely
Amazing.
critical.
Amazing. All right. So from each step to the next, from each cycle to the next cycle, we have a confirmation at the end of it that we're doing well. So information keeps being copied without distortion.
Okay, so now we're going to get into the fun stuff. So everybody, brace yourself.
Wow.
But so this is a cartoon showing a DNA molecule, a naked DNA molecule that it would be the type that you would find in a prokaryotic cell, bacteria. The replication process is a much more involved in eukaryotic cells because DNA is in the form of it's linear DNA that is complex with proteins. And so it's a bit of a more involved process. But the principle, can be, the principles of how that replication proceeds can be readily understood by thinking about replication in bacterial systems. And so the replication begins at a site called the origin of replication. And it's a particular sequence of nucleotides that the cell's machinery recognizes as the site for the origin of replication. And what happens is that at that site, the double helix is locally unwound to expose the two single strands, and they form what is called a replication bubble, right? So you can see that in the second circle is that bubble forming. And that's where the replication is taking place. And over time, that bubble becomes larger and larger until you wind up actually
Splitting it
generating
up.
generating two daughter molecules that then can be released from each other. And the replication is taking place once the bubble opens up in two directions. And so what's called the replication fork, which is the place where the replication is actively taking place. And so replication actually is proceeding in both directions. So when the bubble opens up, you create two replication forks. And so the replication is going in opposite directions around the DNA circle. So it's a highly efficient process because it's going not in a single direction, but simultaneously in two directions.
Wow. And it just drew my attention so much about the origin of replication having a special code that is then recognized.
Yes.
So this to me seems like um, something amazing, because for the replication machinery to work, the molecule itself is telling it where to start, and it just reminds me of communication systems. Like if you're sending something with Morse code, you need to send something at the beginning, just to tell somebody that here I am starting to send a message or something. Or files stored on a hard disk would have a directory service or something that points to the beginning of a file. And without this, you wouldn't know where the file starts or where it ends. So doesn't this on its own, or by its own virtue, is pointing to a structured language? So this is the beginning of the book. If you want to start copying the book, you need to start from there, especially in this amazing structure where DNA is actually circular. So you need to know where to start and where to end. And
Yes.
does this, in your opinion, point that this is a structured language that's
Yes.
lending itself to be read?
Yes, because you could imagine that if the cell's machinery could arbitrarily bind to DNA anywhere and start the replication process, there's no way to regulate it, right? There's absolutely, but by having an origin of replication, you can essentially have proteins that are bound to that origin that will initiate the replication, or will wait until you're at the appropriate point in the cell cycle to begin that replication process. So it helps to regulate the timing of when replication happens, and it allows it to proceed in a orderly manner as opposed to this highly disordered manner. So so to me, there's a very much a logic to it.
Yeah.
Now, what's interesting is that in bacteria, the amount of DNA that has to be replicated is relatively small compared to in a eukaryotic cell. And so we have
Eukaryotes are like animals and plants, right?
yes.
Yeah.
And there's some single-celled eukaryotic organisms as well. But here you have huge amounts of DNA that are partitioned among different chromosomes, where each chromosome corresponds to a large linear piece of DNA. There, in order to have replication occur in a timely fashion, and again in conjunction with the cell cycle, you have to have multiple origins of replication. So when it's time to replicate the DNA, there's multiple origin sites that are along the same DNA molecule. So it's a way of being able to replicate that massive amount of DNA.
So it's like parallel processing in computer systems.
Yes, exactly.
Yeah, and now those origins of replication, if I understood you correctly, those replication starters can only land and bind to that landing pods. If it's a different code, they cannot bind. Is this the case?
Yes. The origin is a precise sequence that again tells the cell's machinery this is where to begin the process.
Wow. So another fingerprint of something that looks like also like irreducible complexity again. Definitely an integrated system that has to be intended to be this way.
Yes.
Yeah. All right.
Now here is we're going to blow up the replication bubble. Now remember, we talked about the idea that the strands of DNA are anti-parallel. They go in the one goes 5' to 3', the other goes 3' to 5'. Well, as it turns out, the direction of DNA replication can only go in the 5', 3' direction. So the template strand is read 3' to 5', and the new DNA strand is laid down in the 5', 3' direction. Now, this creates a complexity.
No, of course.
Yeah, because what happens is at a particular replicating fork, one of the strands has the right orientation, and so DNA replication can begin right away on that strand, and it proceeds in a continuous manner once it starts, and that turns out to be called the leading strand, or the continuous strand. Now, the other strand has the wrong orientation. It's oriented not 3' 5', but 5' 3'. And so that means that what happens is that as the replication proceeds on the continuous strand and the replication bubble gets larger and larger, eventually enough of that other strand is exposed to allow the cell's machinery to get in there and start to do the replication in the opposite direction. And then once it reaches the fork, it has to stop. And then eventually more will be opened up, and then it can actually proceed proceed in that opposite direction. As more opens up, it proceeds in the opposite direction. So that strand is being produced in a discontinuous fashion, where fragments are being produced at a time. And
But,
so
but
that's
how,
called,
those fragments will need to be probably stitched together one way or the other later, right?
oh yes, they will. Eventually once the process is completed, there's an enzyme called DNA ligase that'll go in and kind of join them all together. But this is called the discontinuous strand or the lagging strand, because the replication is lagging behind on that particular strand.
Okay.
So here's another cartoon that helps to see what's happening. So, as the replication fork is opening up, you're going reading the template 3', 5', and the synthesis is going 5' to 3'. On the other strand, because it's the wrong orientation, you're producing those fragments bit by bit by bit, and eventually.
The size of each fragment is like the new opened amount in the fork, and then when it finishes, it starts over again. So it does like this, and then go back. It does like this, and then
and those fragment sizes tend to be uniform. I don't know off the top of my head what the number of base pairs or a number of nucleotides that constitute the fragments, but the biochemist that discovered this was a Japanese biochemist by the name of Okazaki, and so these are called Okazaki fragments sometimes.
Okay.
So there you go. If you're at a Trivial Pursuit party and that question comes up, you'll know the answer.
Yeah, and it's amazing to me that it's just amazing to me the part about gluing them together, because it's again, what if, number one, how does the system know, for God's sake, that it needs to go or one of them goes just straight ahead and the other one, once it stops, because there must be like specialized machinery that recognizes this, right?
Yes, exactly. And so now we're going to take another look at this, where we're going to throw in the proteins that are responsible for the process. And so what happens here is, again, you have the DNA double helix, and there is and this is a simplified description, by the way. This is not exhaustive. I'm simplifying things, but as part of that the machinery that binds to the replication, the origin of replication, there is a helicase protein, and as the name implies, it unwinds the double helix, producing kind of that replication bubble, beginning the replication bubble, generating single-stranded pieces of DNA. Now, single-stranded pieces of DNA are fragile. They're subject to breaking down. So there are these proteins that are called single-stranded binding proteins that bind to help stabilize that single strand so that nothing breaks during the replication process. Now,
amazing.
The enzyme,
so it's protecting them.
Yes. Now the reason
those are the blue blobs.
Yes, the little
blue beads.
Yes, exactly. Now the enzyme that does the replication of DNA is called DNA polymerase, and it actually is responsible for the direction of DNA replication. in the 5 prime 3 prime direction. So it's inherent in the properties of the enzyme and we'll see why that's the case in just a minute. But it goes 5 prime to 3 prime, and it's the same DNA polymerase on both the leading and the lagging strands that's doing the replication. Now it turns out that DNA polymerase is a finicky enzyme, and it won't get the process started unless its demands are met. And it has to have what's called a free 3 prime hydroxyl group before it can start the process. And so what that means is that before replication can proceed, there's another enzyme that has to do its work. This is called an enzyme called a primase. And what it does is it lays down a very short strand of RNA that's about 10 genetic letters in size that is essentially an RNA primer. And once that primer is laid down and it reaches about 10 RNA nucleotides in length, and that RNA will base pair with the DNA, so it forms kind of this mixed or this hybrid duplex, an RNA DNA hybrid. Eventually, when it gets to a long enough length, the DNA polymerase will then add the very first DNA molecule or DNA subunit to the daughter strand, the growing daughter strand at that RNA primer. And this actually happens over and over and over again on the discontinuous strand or the lagging strand. So every time you make a new Okazaki fragment, you've got to lay down a primer first. So it's a very involved process. So then once replication is done, there is an enzyme called an exonuclease that will come in and take out the RNA primer. And as it does it, after it does that, another DNA polymerase that's different from the one that replicates the DNA will come in and fill in that missing piece of DNA. And then an enzyme called a ligase will join that little DNA fragment to the continuous strand. On the lagging strand, every piece of RNA has to be taken out and then replaced by DNA, and then it all has to be joined together by, again, a DNA ligase. And so when that happens now, the replication process has been completed. And so, you can see that this is an incredibly complex process. And I think you have a video that you're going to show us about a computer simulation of this process in action. And so the simulation is amazing because it's showing how rapid the process is going on. But what's remarkable is, in light of how rapid it's occurring, is to think about all these different steps that are taking place.
Absolutely.
That constitutes that process of DNA replication. So, an incredibly complex process that is taking place here.
The thing that strikes me is the fact that the system knows what's happening. It's just completely mind-blowing that it just keeps laying on those primers, and then the copying goes on, especially on the lagging strand, and then it stops, and then a new primer is put so it can start and do another piece. But the system understands that the required result is actually a DNA double strand, so it needs to go back and take off those primers, and then our DNA polymerase friend needs to come and replace them with DNA. But now you have separate strands, and then something else needs to come and glue them together, or I don't know, do this kind of ligation as the ligase guy. So it lends itself to the idea that since these are completely inanimate molecules, they're just molecules, that they have no understanding that the purpose of what's happening is to do a copy, a faithful copy.
Yeah.
So this can only be the case if somebody has designed it. I don't really understand how can anybody look at this unbelievably very intricately engineered process. And the first time I saw one of those computer rendering that we just saw, that you've just seen, it really brings tears to your eyes.
Yes.
Because and when I've showed it when I've shown it to some of the viewers, and actually I sent it to some family members, etc., it's completely shocking. They say, "Wow, those are turbines. Those are machines. Those are this kind of machine can only be the making of an engineer." It's unbelievable, right?
Yeah.
And
now, if you remember to the first episode we did, when we talked about DNA wires, the DNA wire phenomena is actually part of the way in which the replication machinery knows when to begin to start laying down DNA and stop laying down the RNA. So that both the primase and the DNA polymerase have these iron sulfur centers. And what happens is that when the RNA strand being laid down by the primase is long enough, about 10 nucleotides in length, it's long enough now when it's complexed with DNA to actually set up a little mini wire. And so then it's the sending of electrons from the primase to the polymerase that then stop the primase from its work and gets the polymerase started with its process. So there's actually the wire.
They're talking together.
They're talking together through that wire phenomena. Isn't that crazy?
Wow, that's completely crazy. Absolutely, and and do you think that? I think there must be much more miracles in this. That, for example, why is there ligase in the first place? And how does it find the Okazaki fragments? And every time it finds them, and how does it actually really glue the right fragments in the right order to each other? And these are all questions in my mind. Have all of those been solved? Because for sure there is a way. It's not happening by chance every time, because cell division is happening every single moment in our bodies, right?
Yeah. I don't know if anybody has answered those questions. They are the right questions to ask, most certainly. I just don't know if people have explored those questions. But and it may be that nobody really know. We know that this is what happens, but we may not necessarily know how it happens precisely. And I suspect that if we unpack that and develop those answers, I think we would end up seeing a logic, and a rationale to what's going on. Now, one of the things I think people, having seen the video and having heard our description, recognize that this is an unbelievably complex process.
Absolutely.
And one of the things that I learned a number of years ago that keeps me awake at night is that it looks as if this process of DNA replication, from an evolutionary perspective, would have had to evolve 2 times independently to produce an identical system in bacteria and archaea. And this is a phenomena called convergence. And convergence refers to the idea that from an evolutionary perspective, evolution actually evolved independently to the same endpoint, to produce the same kind of system. And we see examples of convergence at the organismal level, at the anatomical and physiological level, and even at the molecular level. And one of the most extreme examples of convergence I know is, again, with regard to DNA replication. Now, why this is problematic is because from an evolutionary perspective, convergence should be actually rare. That when you look at the way Stephen Jay Gould understood the evolutionary process, and his view is shared by many biologists, and that is that essentially evolution consists of a sequence of chance events, and that to be able to replicate those chance events in the exact same way is highly improbable. And each chance event actually uh, has radical ramifications for what happens afterwards. So if you change an event in that sequence of events ever so slightly, the outcome is going to be very different. And so this is how Gould described the evolutionary process, that no finale can be specified at the start. None would ever occur a second time in the same way, because any pathway proceeds through thousands of improbable stages. Alter any early event ever so slightly, and without apparent importance at the time, and evolution cascades into radically different channels. And so his metaphor was that if you could rewind the tape of evolution and let it replay, you'd get a different outcome every time. And so you would not expect something like DNA replication to even evolve a single time, given some of the points you're making, let alone a second time. And if it did, you would never expect it to be, in effect, identical. And yet this is exactly what has been discovered, is that the replication of DNA in eubacteria and archaeobacteria is the same mechanism. The same enzymes are involved. But when we look at the DNA sequences of the genes, there's no way to reconcile those sequences to a shared ancestor. They look as if they essentially have evolved separately into distinct, through two distinct evolutionary pathways. And again, this is completely unexpected when we think about the sheer complexity of DNA replication. A guy by the name of Bill Schaff, who's an origin of life researcher, in a book called Life's Origins, wrote this: because biochemical systems comprise many intricately interlinked pieces, any particular full-blown system can only arise once. Since any complete biochemical system is far too elaborate to have evolved more than once in the history of life, it is safe to assume that the microbes of the primal last common ancestral cell line had the same traits that characterize all its present-day descendants. And so from an evolutionary perspective, you would expect that DNA replication would have evolved in Leuca, and that then as Leuca gave rise to the archaea and the bacteria, it would be the same system. But it looks as if DNA replication must have evolved separately in bacteria and archaea after they diverged from Leuca, which is again absolutely mind-blowing. And so from this standpoint alone, I would argue there's absolutely no way that evolution could explain could explain DNA replication. So it's not only the irreducible complexity of the process, but it's the fact that we see this example of convergence. Something has rigged the system so that the outcome is identical. And this to me looks like the fingerprint of a mind.
So what you're saying is that if we presume Darwinian evolution,
yes,
we will have to solve it by saying that replication evolved independently twice,
yes,
but at the end it is exactly the same system.
Yes.
It went into different paths, but
yep.
At the end of the day, you set up two sets of engineers, one in North America and the other one in Central Asia, and you tell them, build the building of your dreams. They have had no connection to each other, and both of them build two buildings that exactly look like, I don't know, the White House or something, or the pyramids. And it's exactly the same thing at the end of the day. Maybe they use different materials and they they have different paper for their, and they're coming from different places of the world, but they eventually end up building the same two things.
Yes.
And it can, I think then your assumption that those two guys have had no connection to each other has to be false,
Yeah.
and they have had to have the same educator, or they got the same design from the same mind or something, right?
Yes. Yes. Exactly right. And the two quotes that I showed you from Stephen Jay Gould and from Bill Schaff, these are again men that are deeply committed to an evolutionary view. Stephen Jay Gould died a number of years ago, but when he was alive, he was deeply committed to an evolutionary view for life's origin and life's history, and are telling you that you would not, in their statements, that nobody in their reasonable mind would predict that these systems could have evolved separately. They would have had to evolve a single time because they are so elaborate, and yet we see again, this incredible example. Now, to me, and I think you were already hinting at this very strongly. I see convergence as being, not something that you would expect if there was a creator. Years ago, I worked in research and development for a Fortune 500 company, and the company, would constantly remind us to not invent new technologies, but to copy things that other people in the company have already done. So, we were a big enough company that, and so they called it search and reapply. That you would get more credit if you reapplied an existing technology in a new way, maybe, than if you invented a new technology from scratch, because it was highly efficient, right?
Right.
Right.
Of course.
So a designer, is in their right mind is going to reuse the same designs over and over again. So it's something that you would expect from a designer, not something you would expect at all from unguided evolution.
Absolutely. And when you said that what they found was that sequences were different, what is the implication of the difference of those sequences? Is it only just difference in sequences, or even the machinery involved, while it does the same functions, but maybe they're also slightly different to the extent that you can infer, if you're thinking evolutionarily, okay, that they came from different paths and eventually they do the same thing. Is it just sequences, or even the machinery? There are differences in the machinery.
Yeah. There are differences in the machinery as well.
Wow.
But the differences are not substantial in terms of the mechanism of function. That in other words, the machines are different enough that you can't reconcile them through
from an evolutionary process.
Yes.
They cannot be directly traced back to a common single design that branched. So you have to assume that they reach there through two different paths of random mutation and natural selection. But guess what? Wow! At the end of the day, they're both doing the same thing.
Yes. Yes.
Wow. That is the question is now, okay, if this is the case, and the basic rule there is that a contingent process cannot happen independently twice, and you expect the same complex system to emerge at both ends. What is the explanation that the theories that Darwinists would put on the table for that?
I don't really know what the answer would be, other than to say it's just the same selective pressures forced evolution to the same endpoint. But in other words, it's really just hand waving. Or when I've brought this point up, it's like, well, it doesn't surprise me, right? Evolution can repeat itself. It's really it's not, it's a very quick dismissal, I found, without really thinking through exactly what the implication of that insight is.
So it is like the same answer, that there is no other option than evolution. So the evidence that it is evolution is that if it eventually happened, and since it eventually happened, then evolution is presupposed, then we'll just call it convergent evolution and call it a day. This,
Yeah. Yes. Yes. Exactly. Yeah.
stick a name tag on it and we'll just call it that.
Yeah.
Wow.
Yeah.
That's really miserable.
Yeah.
Um,
Well,
yeah, go ahead.
I think you when you interviewed Gunther Beckley, you're telling me that you guys had a long conversation about convergence. And the thing is, once you have the phenomenon of convergence, you can't escape a creator.
Yeah.
So either convergence is indicating, the creator directly designed, biological systems. The language I would use would be, through fiat command, through direct intervention in some way.
Yeah.
Or the creator jimmy rigged evolution so that the endpoint was going to be exactly what the creator intended. So even if you're adopting an evolutionary view of life, when you start having convergence being widespread, and again, this DNA replication example is an extreme example that is, I think, deeply problematic. Even if you want to view life in evolutionary terms, you can't escape a creator, because
Yeah.
the point would be that somehow things have been rigged so that no matter how it evolves, it has to go to this exact endpoint that was intended from the very beginning. So either you have placed the ball at the bottom of the hill, or you've built a ramp so that when you roll the ball down the hill, it goes exactly to that spot. Either way, you've designed and purposed that ball to be, at that precise location at the bottom of the hill, whether it's through the machinery you've built to get it there or by placing it there. So that, to me, is what's very interesting about convergence, is you can't escape a creator once you acknowledge that this is widespread.
Absolutely. And since you have the example of a camera here, so we were discussing the Cambrian explosion, and in the Cambrian explosion, there is suddenly vision, so creatures can see. Just before that, everything was, in terms of what we call vision, not just like being sensitive or responsive to light, but it's actually seeing. So you find those creatures called trilobites that have compounded eyes, like bees. Okay. And I typically have those discussions with evolutionary biologists, and I tell them, guys, how can you get a compounded eye? And they say, okay, look at all those different simpler kinds of eyes. And then I ask them, well, but are those eyes on the lineage of the trilobite? They say no, they are not. So I say, then they are not transitional forms. And I was shocked when Gunther told me that evolutionary theorists say that eyes have evolved 45 times independently, each of them without a precursor. Guys, come on. Now we are looking at what you've shown us, and it's just 2 times, and it's utterly impossible to fathom. And
Yes.
those guys would go for something like eyes and tell you 45 times of convergent evolution. But and the funny thing is that it is like this: may there be eyes, and then suddenly all creatures, new body plans, and there is an eye.
Yeah.
It's a different eye. It's just what you were saying. The eye, it does the function of vision, but this one has, like a single eye. This one has this kind of eye. This kind has that other kind. They're all eyes. You can identify them as eyes. And it's unbelievable because it immediately lends itself to a creator or designer who wanted to have vision on. And there was this other fun example of echolocation.
Yeah.
So bats have echolocation, and some of those whales have echolocation. And obviously, bats and whales When you go to the common ancestors, you'll find so many things in the middle that have nothing like equilocation. So
Yeah.
then they will go again. Conversion, evolution, equilocation
Yeah.
evolved so many times. So it's amazing, really, when somebody is escaping an inevitable explanation, he will then have to go to absurd explanations. I guess
Yeah.
this is,
Yeah.
yeah, this is the only way to be there. All right. So are there any other amazing stuff about replication that we're going to tell us today?
Let me do a couple of quick points, and then we'll bring things to a close here. But one of the things that makes DNA replication so complicated is the idea that you have that DNA replication can only go in the 5' 3' direction, where the DNA polymerase is reading the template strand 3', 5', and replicating the DNA 5', 3', and the anti-parallel orientation of the two strands of DNA to make the double helix.
Mm-hmm.
That's what is creating the complexity. And we did say that there's a reason why you need to have an anti-parallel orientation to the strands, but again, it's this feature that really complicates things. So one thing that you might ask is, could it be, why couldn't you have replication going in both directions, 5', 3', and 3', 5'? That seems like that would solve the problem, completely. And so, in fact, because you see this cumbersome nature, or what it on the surface appears to be this cumbersome nature to DNA replication, many people would say, well, this looks like it's the product of evolution, not, again, something that a creator would produce. But it turns out that we've discovered a reason why DNA replication has to go 5', 3', and why it can't go 3', 5'. And the explanation is a little bit technical. So I'm just going to just make the point, and then trust people, if they really want to dig into the details, they can do that on their own. But if you went 3', 5', the chemistry demands actually a more complex chemical process that involves two reaction steps that would require an enzyme with two active sites that would actually have to be linked together to pull this process off. And so we don't need to look at the details here. Just the top is what it takes to do a 3', 5' elongation versus a 5', 3'. And you can see 5', 3' is a simple single reaction step that is chemically far less complex. So if you actually had replication going in the 3', 5' direction, or you had both possibilities, you would wind up with, again, a very cumbersome system. And then on top of that, you can't get what's called proofreading functions in the DNA polymerase. One of the things that the DNA polymerase does is, as it reads the template strand and starts laying down the nucleotides in the growing daughter strand, every so often it stops and it backs up and it cuts out some of the DNA nucleotides that it laid down, and then it goes back and it repeats that process again. That's called a proofreading function, and it happens more or less randomly, but it's a way to ensure high fidelity replication by just, so it'd be like me knowing that I'm copying a very complex set of sentences and stopping every so often just to make sure that I copied what I had done up to that point correctly before I keep copying,
Wow.
And if I did, I can erase it and then replace the error quickly. So essentially you have that proofreading function, which in and of itself is a very elegant design feature, but you would lose that proofreading function because of the chemical constraints if you went in the 3', 5' direction. There's another reason why you don't want to do a 3' having both directions, because if you're at a replication fork, you've got a primase enzyme going in there to lay down the RNA primer. Then you've got DNA polymerase doing its thing. If you had basically DNA replication going simultaneously in both directions, what would end up happening is you'd have all these enzymes crammed into this really small space, just gunking everything up. So you literally wouldn't have the room available to fit everything into that replication fork to allow for, continuous replication on both strands. So you have to have the discontinuous replication in order to have proofreading and to essentially achieve chemical simplicity to the process.
Does proofreading happen in while the lagging strand is switching gear? Is that the relation?
It happens, as far as I know, it happens randomly. So
Okay.
it just every so often the enzyme will just simply reverse directions and take out.
Why does it need a lagging strand direction to be able to happen?
Because in other words, the chemistry, so that what you're doing is you're laying down the polymerase is 5' 3', and then the exonuclease is going 3', 5' to cut out what you've laid down. But because but in other words, going 5', 3', when you're synthesizing DNA is simpler than going 3', 5' when you synthesize DNA. Likewise, when you're cutting out the strands, that again, it's a simpler process going 3', 5' than to cut it out 5', 3'. So that because DNA has an orientation or a polarity to it, what's chemically true in one direction isn't chemically the same in the other direction. And that makes proofreading impossible, and it makes the 3', 5' addition much more chemically complex. I don't know. This is again very technical, but the point I'm trying to make is that this lagging strand complexity is actually demanded because of the chemical constraints in the process. You have to do it this way. You can't do it any other way. But what's interesting is that the cell has actually, or the cell's machinery that does this replication, actually it's coordinated so that the helicase that is unwinding the DNA doesn't allow the leading strand to get too far ahead of the lagging strand, and so it will slow down to give the lagging strand time to catch up to the leading strand. And the primase is at the just right level in the cell to initiate DNA replication at a rate that's fast enough for the leading strand and lagging strand to be more or less in sync with each other, but not at such a high level that it would allow the lagging strand, or not at a low enough level that the lagging strand can't keep up. So the primase levels in the cell are fine-tuned to keep the coordination between the leading and the lagging strand more or less in sync, so that you don't have a runaway process on one strand that could then create complexities or cause that single strand that's exposed to undergo breakage.
So you mean even the RPM of the helicase is fine-tuned so that it's not too fast for the lagging not to be able to catch up with the leading.
Yes.
It's just right between something that is good for both.
Right. And that the level of primase is that the just right level to allow for the lagging strand to be initiated at a fast enough rate to keep up, but if it was a little bit lower or a little bit more, then you would have problems. So it's at the just right level. So that it's a highly fine. So at first glance, it may look like, why would a creator create something so cumbersome? But then when you realize that there's actually a rationale behind all of this, you suddenly have a system that is probably the most efficient way to do DNA replication, and the simplest way to do replication.
That's again one more example of asking the why question and ending up finding an answer, even when the process um, looks so complicated, to the extent you might at the beginning think that there is no answer.
Yes.
All right. So what else do we have
Well,
about replication?
I pretty much have burned the wick to the bottom of the candle here.
Yeah, I had a question about replication that always I thought is pretty interesting. So you have those nucleotides arranged in a certain way, and then you need to bring nucleotides that are complementary to them to form the daughter. So my question is, how do you end up getting the nucleotides in the sequence that you want? Because the way I imagine it is that there's this huge cloud of nucleotides all over the place. They are produced somehow, and then there is all this mess of Lego pieces that are come in one of four shapes. But at the end of the day, you would want exactly the same sequence. So what if an A comes and we need a T? Does it for each new nucleotide, does it need to try four times and it will succeed one every four? And how does that work?
Yeah, that's a great question, and I'm not sure I know the details mechanistically of how the DNA polymerase enzyme actually is doing that recognition, but I suspect that because as the new daughter strand is being laid down, it's forming it's base pairing with the template strand, and I imagine that the base pairing, so that if an A comes in, it just won't align in the right orientation, probably with the template strand, so that the polymerase reaction can take place. So it probably involves hydrogen bonding, which kind of positions that new nucleotide in the right spot, that then allows the enzyme to operate on it, to attach it to the growing strand. So I suspect that's probably what's going on with the hydrogen bonding interaction. But again, it's obvious that the process has a little bit of slop in it, just because it's a chemical process. It doesn't surprise me. So there will be occasionally a mismatch that takes place. So this is why every so often, periodically, every so often randomly, backing up and taking out what the enzyme laid down ensures that if there was a mismatch, you likely have corrected it or have taken it out, right?
Yeah. Um, I'm wondering if somebody is really doing some research about if there is some sort of, I don't know, um, ontogenetic mechanism or something that is guiding the right nucleotides in the right sequence, one way or the other. So if you have an A, you don't really need to try an A, a C, a G, and then wow, we finally get the T. You
Yeah.
know, because it seems if you need to replicate 3 billion base pairs,
Yeah.
and you need to try actually 12 billion times to get the right thing, I think there is so much for us still to learn about how ligation really happens, how the
Yep.
right pieces find themselves each other, how the right nucleotides would essentially arrive at a probability that is better than 25% or something.
Yeah.
Because given that all of this is chemistry really swimming in water,
Yeah.
it's really amazing that it really happens, and that
Yeah.
organisms are not really, turning into abominations
Yeah.
after 2 minutes.
Yeah. Well, and I mean, you're looking at, to your point, you're looking at highly sophisticated highly sensitive molecular recognition in order to pull off, because the structural differences between the nucleotides isn't that great.
Yeah.
And so you're looking at, again, probably a very sophisticated system of molecular recognition, that has, again, some way of discriminating between those nucleotides in an efficient manner. I just don't know enough about really anything about the structural features of DNA polymerase and how that would then mechanistically contribute to the point that you're raising. Maybe people have done the work and I'm just simply not aware of it. It'll be worth looking for that, looking that up, to see if because that would be, I mean, in and of itself, fascinating to know how that happens.
Yeah, so I think that this matter of the genetic system remains to be so unbelievably amazingly and intricately designed that I believe that there will always be questions raised. Whenever we reach a frontier, we find more questions and spawns more research, and and we find more marvels of the creator there. So that was just replication. What else should we expect in the rest of the central dogma?
Yeah, we're the next episode we'll be talking about transcription, the process of generating RNA, and how that RNA winds up getting processed. And so we see some very fascinating features of transcription that I think point to a creator. That also, again, I would be really interested to hear your perspectives, as a communications engineer and someone who's well versed in information, information structure and in theory. And then we're going to talk about translation, which is, again, an incredibly complex process in terms of how that messenger RNA, once it's produced, makes its way to the ribosome, how the right amino acids get delivered at the right time. So both of them are going to, I think, reveal more marvels of creation and give us more opportunities to see God in biology.
I think this is a great journey, and I've enjoyed every minute of today, and for sure for the remaining installments. And I hope the audience have enjoyed it as much. And everybody, if you have your questions already on the live chat, or you can just put them on the after show, where we'll be jumping to the other side right after this concludes, and you'll find the link in the pin to the live chat. And Faz, this has been another exciting session. I encourage everybody to do further research about the topics that we have seen, and to share it, and to share this episode with your loved ones, and marvel the work of our creator. And thanks a lot, Faz, for being here today.
Oh, thanks, Ahmed. It's always fun.
Thank you very much, and peace be upon you all, and see you again soon.